Okay, thank you very much for coming. We're delighted for today's Museo de Ciencias Naturales seminar to have Maria Ikonomopoulou, the head of the Translational Venomics Lab at the IMDEA Food Institute in Canto Blanco here in Madrid. So Maria did her degree in agricultural technology at the Western Macedonian Technical Education Institute in Greece. She then did an MSc in zoology at the University of Tasmania, and then stayed in Australia to do a PhD in biomedical sciences at the University of Queensland. Then Maria also did 2 postdocs in Australia and obtained an associate professor position at the University of Queensland. And she now holds a senior Comunidad de Madrid Talento Fellowship at Canto Blanco. And in her lab, the research focuses on the amino acids that are active in venom derivatives and their application to biomedicine. And that's what Maria is going to talk to us about today. So thank you very much.
Thank you, Rob, for the very nice introduction, and many thanks to Ida for the invitation to be here with you today and present our work. Great. So I'm going to talk a little bit more about the anti-cancer properties of animal venoms and their potentials as drug candidates. So probably most of you already know some of the venomous species like the spiders, the scorpions, the snakes. But I also like to highlight that there are other venomous animals that are less well known, like the bullet ant that is in the bottom part, as well as the platypus, the male platypus, that only the males, as I said, has venom. And it's actually, we don't have many information because it's endangered as well species in Australia, is protected. So, but there are cases that cattle that accidentally, they had been envenomated, they died. So it's a very powerful venom. And then of course we have the river worm, that also a worm that Ida likes. So from the aquatic environment, there is the jellyfish, there are the man-of-war, sea snakes, sea anemones, the cone snails. I think Rafa and his group will be able to tell you more about it. But I always like to highlight it because I'm fascinated about the cone snail, that I see that when it just envenomates the fish, it just instantly paralyzed. And it paralyzed for a prolonged period. So it gives enough time to the cone snail to enjoy its meal. So it's a fascinating and terrifying, I think, for me, side. But it also shows the potentials and how active and potent this venom is. So the venomous animals have evolved throughout the years at different, not all of them at the same time point, but along with their evolution also, the venom has evolved. And I like to highlight that the venom is not just one thing, it's just a complex mixture. So it's got salts, got enzymes, small compounds, peptides, and big proteins, especially the snakes that they have. So, and it's not all of them, they are just targeting one thing. So there are just different properties and that the venoms animals have studied. And of course there are others like the jellyfish that we have less information now. So merely the scientists have barely studied just a few of them and their components. So there's still a lot of work to do and a lot to explore and focus on. So some of the applications of the animal venoms, I mean, they have a very wide pharmacological properties. They're just ranging, as you see, from some of them that I've listed, from analgesic, antimicrobial, antiparasitic, as well as anti-cancer that we are focusing on, and metabolic. But also they are used as tools. For example, we know the case of the chlorotoxin, which is coming from is a peptide that's coming from the Israeli scorpion. We know that because of its specificity towards glioblastoma cells, they are actually explored now in clinical phases. I think it's 2 as a paint, as a tool, so the surgeons, when they are doing the surgery, they can easily take the glioblastoma cells. And there are others like bee venom components that are also being used as carriers to transfer small molecules or even peptides into cross the blood-brain barriers. In the cosmetic industry, especially in a lot of Asian countries, they're used as creams, as products, as a skin whitening or depigmentation skin products, and also the spiders. Obviously, the most well-known is the agrochemical properties, especially from the spiders, because spiders are, as we say, the master kills of insects. So it's not surprisingly that there is a bioinsecticide developed and used and approved in EPA that is coming from a funnel web spider. And now they're trying to expand in Europe. So, and also, so not people get scared when we talk about venom, is the dose that makes the poison. It's not that all the venomous animals kill. So there are currently 11 approved FDA and EMA drugs, venom-derived drugs. As you can see from the list, most of them are to treat hypertension, thrombosis, acute coronary syndrome, against chronic pain, diabetes, as well as anticoagulant. And the majority comes from the snakes. The reason for that is that when all this research started, blooming, people were focusing on snake venoms for the obvious reason that snakes yield a lot of venom. So it was easier to work with the venom, to characterize it for the properties, study it, and eventually to have the first drugs. But now, of course, with all the evolution and the omics exploration, we are anticipating that there might be more venom-derived drugs coming from other species like, for example, centipedes or even river worms. So there are, of course, many others that they are different stages of preclinical and clinical development, and from all kinds of different animals for treating cancer or for other different disease. They're investigated for immunomodulating properties, against stroke, and so on. And even, I think, sea anemones, they are investigating it for psoriasis, and they have already passed clinical phase 2. And in our lab, as I said, we're focusing on cancer, more specifically for melanoma. Why? Because melanoma is the main skin cancer causing worldwide fatalities. And when I started this work in Australia, it's a big problem. So the main mutation is the BRAF that you can see, and this is the focus also for our studies. And luckily, at early stages, most of it is treatable. You can just remove it. But although there are 10% of patients that the cancer relapse. But the problem is when it's advanced, when it's metastasized, it's lethal. And basically, there aren't really good solutions for all patients, unfortunately. So this is very briefly to say about the pathway and different inhibitors that they're targeting in order to stop the proliferation of the melanoma cells. So the current approaches at the beginning was monotherapy, BRAF inhibitors and MEK inhibitors. Later on is targeted approaches where you combine them, as well as immunotherapy is very promising, and combined also immunotherapy. Now they're more into triple therapy, so you combine targeted approaches with immunotherapy. And the problem is some of the challenges that the clinicians facing today with these treatments is that a lot of patients are unresponsive. It's not like a magic drug for everyone, unfortunately. There is a lot of high toxicity rates and a lot of side effects, especially with immunotherapy. And what is happening in many cases with cancer patients, they develop cancer resistance. So then the cancer comes back. And so there is still a lot of room for new approaches to treat melanoma. And that is just our platform. So we can start with the whole venom, or it makes our life easier, the omics, so we can get already the compounds that we're interested in investigating. And something that I really want to incorporate in my research is artificial intelligence as well. And then we want to find a venom that has specific anti-cancer properties, mainly without killing the healthy cells. So this is our platform. So I was saying that we're looking for venoms that specifically target the cancer cells, and obviously that they have minimum to no effect in the healthy cells. And then when we find this ideal venom, we go through chromatography in order to identify the peak or the peaks that they are responsible for the biological activity of the whole venom. And then obviously there are a few more steps of purification of the fractions to get the really nice, very pure fraction. And then you can send it, hopefully, if it's a peptide, especially for sequencing. And then when we get the sequence, we can obviously produce it. We can do it recombinantly in bacteria or in yeast. Many labs are doing that. And we're more interested in the synthetic part. And then we can have enough quantities to characterize it in detail for, in our case, and in this example, for the anti-cancer properties of the venom, get information about this mechanism of action. We're utilizing a lot of omics, proteomics, RNA-seq, the metabolic profiling of the venoms. And then if we don't have the structure, we aim to get it through NMR, and then we can do structural activity relationship studies, hope in combination with medicinal chemistry in order to have better and safe and enhanced analogs. And of course, we want to validate it in the appropriate animal venoms to make sure that they are active. So the challenge is, is that back? That went back. So I want to introduce you just to bring two examples that we have been working for a while in our lab. One is the gomessin peptides that they come from the spiders, and you might know the one that is the most famous, the agom, as I will call it from now on. That this comes from the hemocytes of the South American megaloph spider. And it's 18 amino acids long. As you can see also from the structure below is a vita hairpin. It's got 4 cysteines, 2 disulfide bonds. It's a pore-forming peptide, and it has been studied for a range of properties for antimicrobial, antiparasitic, antifungal, and of course, anti-cancer. So surprisingly, we found another almost identical peptide from the Australian funnel web spider, not from the hemocytes, but actually from the transcriptome analysis of the venom gland. And it's still 18 amino acids long. And as you can see, the only difference is just these 2 amino acids that they have, that you can see below. And we studied it, and we saw that they do have antibacterial and anti-cancer properties. And then we wanted to dig in a little bit more to find out if just the 2 amino acids or just sometimes 1 amino acid can make a difference in the biological activity of the peptide. So we synthesize a wide different ranges of amino acids based on the differences of those 2 peptides that we were studying. One, we also find it naturally in the spider of another spider, the Ikonomopoulou, which is this one. And then the others, we just did some alanine mutations that they were changing the hydrophobicity and the electrostatic potential to see if there were other factors and cofactors that they might interfere in this process. So then we tested all these peptides in the melanoma cells, the MM96 cells, and in the human healthy fibroblasts, the NFF. And what we found is that most of our peptides, they seems to be very specific towards melanoma, with minimum to no effect towards the healthy cells. So this is like a therapeutic asset that we were really after, and we were very excited back then on that part. And we saw, though, that only just alanine substitutions, that they actually made it, I think, less hydrophobic, the peptide, they just abolished even the activity, even in the melanoma cells. So this is for the chemist, the people that they like chemistry a lot. We did characterize it in detail. I'm not presenting here the cell signaling pathways that they were involved and so on to get an information about the mechanism of action. But we validated also, that's what it shows in animals, in mice, in xenograft. And we saw that when we treated the mice just a little bit over a week, every 2 days with the Australian follow-up gomessin peptides, it reduced the tumor progression as well. So there was a validation in vivo as well. And we coupled these results with another independent animal model, the zebrafish. I don't know if you are aware of this animal. Well, you probably are. But I wanted to say in the context of cancer, how we're using it is because the embryos is transparent the first 10 days, and that allows us, to be able to visualize and is not immunocompetent. So it allows us to inject the human cells, and then when we label it, we can actually quantify the proliferation of melanoma cells basically see that there is a reduction in the proliferation ratio in the embryos that they were treated, in the zebrafish embryos treated with both of the gomecin peptides. And this is work that is happening in the lab of Laura Sanchez and Pablo Cabeza, her postdoc in Santiago de Compostela University. So we were super excited that, and other colleagues independently validated our results. And now we're following it up, getting more into the factors that they affect the cytotoxicity of this peptide, like cholesterol and lipids. And because we know that they have a specific domain that shows it binds into cholesterol, that's some work and molecular characterization that we are currently doing now. And this is a work from Evelyn. Evelyn works with artificial membranes, and she's using this probe, the RH421, and she gets information about the binding, the indirect binding of our peptides, in this case of the Brazilian gomecin, with the different ratios of phospholipids and cholesterol that they usually could be present in a cancerous cell membrane. So she found basically in this one that this combination that is with phosphatidylcholine, phosphatidylserine, and cholesterol increases the binding of gomecin, of agom-gomecin in these membranes. She also found that cholesterol seems to reduce, the more cholesterol there is in the cells potentially, then the less disruptive effect the gomecin causes in the membrane, according to her studies. And we saw similar results, more or less, with the Australian gomecin peptide, and similarly for the cholesterol. So just some of the conclusions here for the work that I briefly mentioned and some that I haven't, is that the gomecin does not bind, the agom does not bind to or disrupt these specific phospholipids, but requires them to bind and to disrupt the membranes. Cholesterol does not necessarily reduce the binding to the membranes, but disrupts the reduction. And we're coupling this with molecular work to validate it, because that was artificial membranes, to see if we can find a direct link with the cancer cells that we're using. The high-GOM shows weak binding to the phosphocholine, shows very strong binding and disruption in the membranes with phosphatidylserine, and cholesterol slightly increases the binding of the membrane but reduces disruption. And then we saw that basically cholesterol is a buffer for the cytotoxicity of the melanoma cells. So I'm going to talk about another interesting peptide that we have been studying, that is coming from an octopus, actually, species, the sand octopus species from Australia. And this is a very typical tachykinin peptide, basically, that I'm going to talk now. So we tested this peptide as well as variants that our collaborator Brian Fry had found from different marine species, but they were very similar in the sequences, just a few amino acids different. And we tested them in our melanoma cells, and we saw that there was one specifically that seems to reduce the proliferation ratio of the melanoma cells. So because this peptide, as you see the sequence that is OctPep1, it had to have a methionine at the end, so it looked like it was losing the activity fast, and it was a little bit difficult to work on. We synthesized an analog, just to, putting isoleucine at the end, and instead of acid, amino acid, and a pyroglutamate in front. And then we saw that even though the peptide was more stable, as we observed, then it was losing more, it was less cytotoxic than the native peptide. So we tested this peptide, and again, we saw that it was more specifically towards the melanoma cells, not affecting the healthy cells. And we validated, that they have similar properties in a range of melanoma BRAF cells of the same mutation. But with different mutations, it didn't have any effect. So what was interesting for this peptide, again, was found from the transcriptome of the venom gland of the octopus sand species. And as you can see, it's 12 amino acids. It's linear. It's not it doesn't have any disulfide bonds as the gomecin. It's not as cytotoxic and potent as the gomecin peptide. And then it has like this domain that you see in red that is very typical of tachykinin peptides. And these tachykinin peptides are found equal in vertebrate and invertebrate species. And they have been involved in different functions, including inflammation and some reports in antiproliferative capacity. We also saw that it's unstructural and disoriented in water. In the presence of a TFA, I think 50%, it serves sort of a little bit of alpha, adopts an alpha helical structure. So we tested it for its, characterized it for its antiproliferative capacity, and we saw that it didn't lead actually to cell death as gomecins did, and it didn't stop the cell cycle arrest. It didn't cause any cell cycle arrest to see if it's another mechanism that is acting that is more common in the cytotoxic peptides. But we did see that then we studied its metabolism because metabolism is a lot affected in melanoma and in cancerous cells. And we did show that it affects a lot of the parameters that are responsible for the mitochondrial respiration. But it didn't have any effect on the glycolytic parameters. So then we did proteomics and RNA-seq, and we saw that this is one of the affected pathways, just to say that this is one important pathway for the proliferation of cancerous cells. So we focus on this. And then because we saw that it had this domain that I mentioned that is specific for the tachykinin peptides, we saw if this potential binding to a tachykinin receptor or neurokinin receptor is the same, had any effect in its properties. So we found out that in the melanoma cells that we used, they do express the neurokinin or the tachykinin receptors, but at different levels. So, and then we saw that if we added the peptide, it didn't cause any changes in a protein or mRNA level to indicate that this receptor might be involved in its biological action. So the full test for this one was when we knock out this receptor, and we knock out the NK1 receptor, which is the main receptor in melanoma, and then we saw that it didn't have any effect. It didn't affect the viability of the peptide. And then I have some more analogs here that I'm going to talk now. So we thought there is maybe another reason, maybe some structural conformation that it might have that it can lead us to its biological activity. So we did molecular modeling with Gonzalo from our institute, the bioinformatician, and he found that it adopts this vita helix structure conformation that the other variants that we had tested and they weren't active didn't have, at least not all of them. So we created a panel of analogs basically based on a hypothesis, which is the active part. Is it the C terminus? Is it the N terminus? What is the importance of hydrophobicity? Or also what is the position of the charges? And of course, still if NK1 receptor has a role, and the polyproline conformation that we observed from the molecular modeling. And what we did find out, basically with viability and other molecular experiments, just the take-home message, is that the C-terminal was the active part. But the N-terminal seems to be equally important in the selectivity of our peptide towards melanoma cells. That's the take-home message from here. And then, as I say, we validated that also with reactive oxygen species, with seahorse data, which is metabolic data. And then we tested it also in mice. And then we saw that after over 2 weeks, when the mice, the nude mice, that they were treated, that they had melanoma cells, melanoma tumors, and they were treated with the OCT-PEP, had a much slower rate of progression of the tumor in comparison to the vehicle controls. And again, we validated the same data with the zebrafish xenograph model, and we saw similarly that the zebrafish that they were treated with the OCT-PEP had less melanoma cells. And the conclusion is that we have this peptide that is not cytotoxic because it doesn't kill the melanoma cells, basically it just stops the proliferation of the melanoma cells, which is interesting. And it's very specific towards a specific mutation, the main mutation of melanoma. It does seem to increase the reactive oxygen species, but it doesn't complete totally the effect, the metabolic effect in the mitochondria. So it seems to be some inefficient, as we say, oxidative phosphorylation coupling. And then I'm not going to talk about mitochondria clustering or where it accumulates, but it enters the cells as we saw, and we saw the effect through the omics data. So it's a weak, as we say, PI3K AKT-mTOR inhibitor, one of the main proliferation pathways in cancer cells. This proliferative capacity is not related to the neurokinin receptors, to the tachykinin receptors that we were initially hypothesized based on its sequence. But it seems that, I haven't talked, we saw that because it's not strong enough probably to be used as a drug potentially in the future by itself, but maybe can be used in combination with other approaches. So one of the combination drugs that we tested was mTOR inhibitors and ERK inhibitors, and we found out that with these specific inhibitors, it synergizes and is very potent. So that could be a potential future use. And of course, it diminishes the progression of the tumor into independent animal models. And oops, that's it. And this is always my most clustering slide because all the work is not one person or one team. So there's a lot of collaboration, and I think it's keep on going because we're trying to follow up these peptides, and we're getting really good data with new collaborations and yeah. So thank you. Microphones.
Hey, thank you very much, Maria. So are there any questions?
Hi, Maria, this is Sergi Lavada from the museum. I was just wondering, very interesting talk, very far apart from what we're doing. But I was just wondering whether you have any collaboration with companies like, for instance, PharmaMar, that work with anti-cancer also, and they do test some particular organisms, and you could potentially collaborate with them. Are you collaborating with them?
So we are starting now, especially with some candidates. We have been approached for another project for Sorry. Sorry. So yeah, because that is the ultimate thing, to collaborate more with the companies. So we're discussions and establishing collaborative agreements with that. And one of our, actually, because this is the octopus peptide is part of now of the national plan that we have a collaborator that he works for PharmaMar, although probably as a clinician, he was part in its capability as a clinician at that point, not as PharmaMar employee. Mm-hmm. Thank you.
More questions? I've got one. So you've shown that there's venom in a lot of different organisms from different taxonomic groups, and it works in different ways. So how do you find the candidate organism or compound to start testing for a particular medical application? Is it just, I mean, there's some mechanisms, I guess, that or is it sort of a lucky guess?
Yes, that's a good question. It's a lucky guess and a lot of high throughput screening, testing a lot of venoms and a lot of compounds in the assays that we are looking at. But that's why we want to incorporate artificial intelligence, because that can save us this step of going sometimes blindly, and then you can go more targeted approaches. So yeah, thank you.
And then, I guess, following on from what you've shown us, there's a long process. You mentioned, for example, that the octopus venom here would be a candidate for using with other approaches. So because of the different sort of mechanisms you find, the way that these venoms work, it must be a long process sort of working out the biomedical applications, in what combinations you would use them, how you would actually produce them. What sort of is the time between you first? How much time would there be likely to be between you first testing something as a candidate and it going onto the market?
Going to the market, I thought you were going to tell me and get a good idea of how it works. So, I mean, yeah, going to the market, they always say, unfortunately, even now in a pharmaceutical area, it still takes 20 years. I mean, big companies, they can reduce it 5 years maybe, but it still is a very long process. And because it's all the safety tests and to make sure you address in the process, to make sure you have a safe and effective drug, basically. And then because the ultimate test is when it gets to clinical trials. I mean, you can get fantastic data in the lab, and then you go to humans and it's not working, or it's not working the way you would like to. So that's why it's a very long process. Unless you go to cosmetics, probably that's an alternative.
Hi, Maria. This is Ana Riesgo. I work very tangentially with venoms through IDA. And I have a question for you, because I mostly work on sponges, which are not venomous, or we don't think they are. We have suspicions, but we don't think they are. But we are finding lots of venom-like peptides. Have you ever tested anything that was not venomous and it turned out to have something potentially useful for you?
No, but I'm happy to test different and new secretions from different species, because you never really know unless you really test it. So,
because one of the things that in many of our grant proposals we are almost forced to say is that sponges never have cancer, so they have really potent blah 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 blah. It's all bullshit. But maybe there's something in there.
Yeah, I wouldn't be surprised. I mean, it's always test and for example, like in the Australian funnel web that comes the comestin peptide that I talk, is the same spider that Vesteron, the company that they have the first bioinsecticide that I said, they have also found it from the same spider. And then our collaborator Glenn from Australia has found a peptide specific for anti-stroke, and he has developed a whole company based on it. So the same spider, different peptides from the same venom with different, very distinct and powerful biological properties. So I think that's my bright example that I like to highlight. Thank you.
Hi, Maria. Very nice talk as always. I had a question about the artificial intelligence you were mentioning. So I assume you want to use those techniques to try to look at potential functions or targets or something like that. So is there a particular difference in the melanoma cells that differs from the healthy cells that you can target? Like, they express more certain channels or something that you can look for compounds that target those?
I mean, I don't know, but we're doing a lot of lipidomics because we have the hypothesis that lipids plays a role that I talk. So we think that the cancer cell membranes, they have a different lipid composition from the healthy, from the non-transformed cells. And these interactions that occurred with the peptide and the lipids might have a role in the biological activity. So the artificial intelligence is something that we would like to see for screening different databases, and it still is an idea in development for finding new targets, or based on the available databases that they are, trying to find also new drug candidates on this. So anyone with bioinformatics skills on this field, please talk to me. Thank you, Ida.
And any more questions in the room? Okay, well, in that case, thank you very much, Maria, for a very thought-provoking presentation. And